Hello, my name is Santiago Lamas and I work as a group leader at the Centro de Biología Molecular Cell Show in Madrid, Spain. In this video, we would like to present the highlights of our work. Renal tubule CPT1A overexpression protects from kidney fibrosis by restoring mitochondrial homeostasis, recently published in the Journal of Clinical Investigation. This work is part of a collaborative effort among Spanish and American laboratories and has been supported by diverse funding agencies, to all of which I am very grateful. Our main findings relate to kidney fibrosis, which is a major clinical and biomedical challenge, as well as a pathology underlying most conditions leading to chronic kidney disease. By using experimental models of kidney damage, we have found that kidney fibrosis is closely related to metabolic failure. However, when fatty acid oxidation is enhanced, as we observed in mice overexpressing the enzyme carnitine palmitoyltransferase type 1, we found that mitochondrial homeostasis and bioenergetics was restored, thus preventing from kidney fibrosis. Consistent with these findings, we found that patients with chronic kidney disease also showed diminished capacity for fatty acid oxidation. To lead you through all these details, I would like to present the first author of this work, Verónica Miguel.
Renal fibrosis consists in the excessive accumulation of extracellular matrix proteins, mainly collagens and fibronectin, in the interstitial space, replacing cellular components. Tubular damage is the driver of kidney fibrosis, which involves alteration in mitochondrial metabolism. They are highly energy-demanding cells which mainly obtain ATP from fatty acid oxidation. When these cells suffer a persistent chronic injury, fatty acid oxidation is shut down. TGF-beta, the major profibrotic cytokine, inhibits fatty acid oxidation in these cells through the depletion of PGC1-alpha and PPAR-alpha, which regulate the expression of fatty acid oxidation-related enzymes. This results in energy depletion, cellular death, and fibrosis. CPT1A is the rate-limited enzyme in fatty acid oxidation. It acts as a carnitine shuttle involved in the transport of long-chain fatty acids. It is located in the outer mitochondrial membrane and allows acyl-CoA carnitine molecules to reach the mitochondrial matrix, where they become oxidized through the 3-carboxylic acid cycle to produce ATP. Hence, we wonder if overcoming the reduction in fatty acid oxidation could result in protection from kidney fibrosis. To address this question, we generate an inducible conditional transgenic mouse model for CPT1A overexpression, specifically in the epithelial cells of the kidney after doxycycline administration. These mice presented a 10-fold increase in CPT1A expression, specifically in the kidney, which was reflected in a 2-fold increase in renal tissue fatty acid oxidation rate. To study if this CPT1A overexpression could prevent from kidney fibrosis, we performed 3 different models of chronic kidney disease in these mice: the unilateral ureteral obstruction, the folic acid-induced nephropathy, and the adenine-induced renal injury, obtaining consistently similar results in all of them. Histological analysis by using the collagen staining reagent Sirius Red reveals that the percentage of fibrotic area was lower in CPT1A non-kin kidneys in the folic acid model. Disruption of organ tissue architecture associated to kidney fibrosis leads to renal function impairment. Common parameters indicative of kidney function: blood urea nitrogen, BUN, and serum creatinine were increased in animals injected with folic acid, but this variation was blunt in mice overexpressing CPT1A. Consistently, these mice exhibit reduced abundance in the expression of the fibrosis markers fibronectin and smooth muscle actin after folic acid-induced nephropathy. This protective effect has also an impact on mitochondrial morphological alteration and fatty acid oxidation defect induced by fibrosis in the kidney. Thus, morphological alteration in mitochondria induced by folic acid, including reduced number, fragmentation, and size, were significantly abrogated in renal epithelial cells overexpressing CPT1A. The decreased fatty acid oxidation rate and ATP content associated with kidney damage were also prevented in the kidneys of CPT1A knock-in mice. To gain insight about the quantitative metabolic changes at cellular level, we examine oxygen consumption rate, OCR, of primary renal tubular epithelial cells isolated from kidneys from CPT1A knock-in mice. We observed that basal and maximum oxygen consumption rate associated with palmitate consumption were markedly higher in cells isolated from CPT1A knock-in mice compared to the ones isolated from wild-type mice. In addition, we examined the effect of TGF-beta on renal epithelial cell metabolism. Cells treated with TGF-beta show a reduction in palmitate-induced elevation in OCR, indicating a low activity of fatty acid metabolism. CPT1A overexpression also prevented TGF-beta-induced bioenergetic derangement. By analyzing the expression of fibrosis markers, we observed that enhancing renal epithelial fatty acid oxidation exerted a protective role in TGF-beta-induced dedifferentiation. The inflammatory response was also decreased in these mice. Thus, kidneys from CPT1A knock-in mice presented reduced renal macrophage infiltration with a visible effect on the M1 population in the folic acid-induced nephropathy model. To investigate if restoration of fatty acid oxidation gain of function after injury could also protect from kidney damage, we start doxycycline administration after folic acid injection. We observed that the fibrotic area as well as the renal function impairment were significantly reduced in the CPT1A knock-in model. The expression of the profibrotic markers collagen 1 and fibronectin was also significantly reduced in the CPT1A knock-in mice. Tubular epithelial cells from CPT1A knock-in mice also exhibit a more favorable mitochondrial bioenergetic profile after damage, which was reflected by a higher ATP production rate and basal and maximum OCRs. This data reinforce the potential of fatty acid oxidation gain of function to combat kidney fibrosis even when renal damage has already been initiated. To explore the importance of fatty acid oxidation in a clinical context, we analyzed two cohorts of chronic kidney disease patients. We found that those with a lower glomerular filtration rate, GFR, show increased accumulation of short and medium-chain acylcarnitines and decreased CPT1A expression levels. These results underscore the relevance of reduced fatty acid oxidation in chronic kidney disease and support the importance of this gain of function to combat kidney fibrosis as demonstrated by the phenotype of the knock-in mice. In conclusion, overexpression of CPT1A impedes tubular epithelial renal cell dedifferentiation and deters a pro-inflammatory and pro-fibrotic phenotype by restoring mitochondrial function.